Good mini break, mid-semester break. We have 3 more weeks, and then we have a real spring break. So just 3 weeks, we can do it. So today we're going to finish talking about the somatosensory system and start the visual system. So here's our outline for today. I don't need to read it. You've got it in front of you for future reference. So let's review the pathways. This is anatomy and physiology combined. So let's talk about the pain and temperature sense modalities of the somatosensory system. Pain receptors are the free nerve endings in the skin and in the visceral organs. The receptors for pain are things like acid, ATP, and adenosine that is released from damaged cells. Those are some of the receptor molecules. Those are some of the chemicals that are detected in the transformation of chemical information into pain information. So let's just take temperature, for example. There's a receptor called TRPV1, TRPV1, you don't have to know the name of it, that is a heat-sensitive ion channel. It's also a capsaicin-sensitive ion channel, so it's activated both by hot spice and by hot temperatures. And it's an ion channel that is monovalent cation permeable, so it depolarizes the nerve endings and causes it to fire action potentials. Okay, that's an example of a temperature sensing. We have ones that are sensitive to cold that are also activated by menthol. We have ones that are activated by ATP that are more strictly pain, not temperature. Anyway, receptors in the nerve endings triggers action potentials that propagate up the nerve. The nerves, all of these nerves have their cell bodies in the dorsal root ganglion. So that's a label that you should put on your handout, because that's a name to know, dorsal root ganglion. And the axons of those cells enter the spinal cord at the dorsal horn. The dorsal horn is not the same as the dorsal root. In the dorsal horn, the pain and temperature sensitive neurons that we've been talking about make a synapse onto the secondary cell. And the axons of the secondary cell cross the gray matter and now go up. Now we're changing orientation here. Now go up the spinal cord in the anterolateral column. This is a pretty bad cartoon of the anterolateral column, so I will make a better cartoon of the anterolateral column, but it's got the name right there. So this is white matter, a white matter tract in the anterior lateral area. It's right about here, not way out here. And those axons of the secondary neurons, white matter myelinated axons, go up the spinal cord, go through the brain stem, and form their synapse in the thalamus. In the contralateral thalamus, because we have crossed over. So the information carried in the anterolateral column of the spinal cord, carrying pain and temperature information, is contralateral. It's carrying information about the contralateral side of the body because there's been a crossover. Contralateral thalamus, processing integration of that information. And then the third order synapse, the third order neuron, synapses here, third order neuron makes synapses in the somatosensory cortex, which we will unpack later today. The touch, pressure, and position pathway has some similarities and differences. Similarities, there are receptors, but they aren't receptors for temperature or for painful stimuli like acid or ATP. They're formed by those touch and pressure are transduced by mechanosensitive or stretch-activated ion channels in those corpuscles that we talked about that cause their stretch-activated cation channels. Cations flow in, depolarization, action potentials propagating in the axons, cell bodies in the dorsal root ganglion, enters the dorsal horn. So I've tried to illustrate some similarities and some differences. No synapse in the dorsal horn. The axons go up the ipsilateral dorsal column. So this is the dorsal column of the spinal cord. This is the ipsilateral dorsal column here. And these axons, the primary afferents, go up the spinal cord and form their first synapse in the brainstem, in the medulla. The second order neuron crosses over. But the crossover is in the medulla and the pons, not in the spinal cord. So information in the dorsal column is carrying information about the ipsilateral side of the body for touch and pressure. Crosses over in the brainstem, synapses in the thalamus, synapses in the somatosensory cortex. Okay, so this was all just review. Oh, proprioception, position sense. The sensory organs are in the muscle spindles, so they are sensing muscle stretch and tension. And some of that you'll see, I'm emphasizing some of that proprioception information from the muscle spindles goes up the ipsilateral dorsal column along with touch and pressure information. So you've heard about the dermatomes. The idea here is that our skin, that there's a section of our skin, the T10 section around our belly button. The sensory neurons that innervate this skin, whether it's for touch, pressure, pain, or temperature, those neurons enter the spinal cord at a specific level. The L4 neurons enter the spinal cord at the lumbar 4 level. So the dermatomes are really define what spinal cord level the neurons enter the spinal cord. Different dermatomes have different entry into the spinal cord and then go to the brain. And different dermatomes have different sensory receptive fields. So you may know that the receptive field of the lips and face entering either, so the lips and face are not entering the spinal cord. V1, V2, and V3, this is not V, this is Roman numeral 5. This is the trigeminal cranial nerve, and these are the 3 branches of the trigeminal cranial nerve. So this is not a spinal nerve, this is a cranial nerve, but the concepts are the same. So, at least as far as somatosensation are concerned. The V3 dermatome has a very fine receptive field. The neurons that innervate the skin, or the lip, have a very small receptive field, and that receptive field is sharpened by lateral inhibition. I'm not going to go through that again. The area of lumbar 2 or lumbar 3 has much bigger receptive fields. So let's play a little game and demonstrate receptive fields. I need a volunteer. So what I have here is a probe with 2 points. They're not sharp. I will not cause pain. I'm only going to activate touch and pressure, I promise. If I start to cause pain, you let me know. That's not the goal. And so I can have these points very close together, maybe 1 millimeter apart, or I don't know how many millimeters that is. 10 millimeters? I don't know. 15 millimeters? I could get a ruler in. Anybody got a ruler? A ruler. Excellent. Okay. So this is in inches. Okay, we'll have to be in inches. So this is 5.5 inches apart. And this is 1 millimeter apart. Okay? So what I'm going to do is I'm going to pick a fine dermatome. The finest ones would be in the lips, but I don't want to put this on Thalia's lips. So let's use a fingertip. So Thalia, I want you to close your eyes and just hold your finger out here. So I'm gonna slowly increase the distance, and you tell me when you can unambiguously and honestly, please, detect 2 stimuli. Because right now, I think 1 millimeter is going to feel like 1 stimulus. That's within the range of her receptive field. But let's see. So, do you feel 1 or 2?
1.
Okay.
Two.
Okay, so okay, I'm going to try to go back. Okay.
That's ambiguous, but I guess 1.
Okay, let's try again.
1. 2.
So that's approximately her receptive field, her 2-point discrimination receptive field. So that is, let's just call it 1/8 of an inch. Somebody calculate that in millimeters. Go ahead. Use your phone and turn 1/8 of an inch into millimeters. I can do it. You do it while I do the next test. Do you know what 1/8 of an inch is in millimeters? 3.175? Okay, 3 millimeters. Okay, let's do the same test on the back of Thalia's neck. So this is the C3 dermatome. So same thing. Honestly, tell me when you can feel 2.
1. 1. 1. Probably 1. 1. 1. Probably 1. 1. 1. I'd say 2.
So somebody calculate what 1 3/4 inches is in millimeters. 34. 44. So a factor of 10. More than a factor of 10 differential. And we're not even going to the most, if I were to do her lower back or the back of her leg, it would be out here like this. You can't tell the difference between this distance and this distance on the lower back. Thank you. That's receptive field, and caused by the innervation of the skin of the sensory neurons and lateral inhibition. Okay, that was fun. Let's talk about the other pathway for proprioception. So I don't think I got to this on last week, a week ago. So this is new. This is the spinocerebellar tract. Now I could do this rough, or I could do this smooth. I'm going to do it smooth. I'm going to make it simpler. I'm going to make it simple. But I'm gonna tell you the whole story, and then I'm gonna tell you what you don't need to know. Okay? So this is proprioception, but this is proprioception that's going to the cerebellum, not to the cerebral cortex. So it's not, at least not directly. It's going first to the cerebellum, and that's why it's called the spinocerebellar tract, from the spine to the cerebellum. Proprioception, so it's got the same muscle spindles are sending information up the spinocerebellar tract. So here are sections of the spinal cord: sacral, lumbar, cervical. Okay, so this is going up the spinal cord, and this is now going up through the brainstem. So it's got a little bit more detail than the previous cartoon. One pathway for the spinocerebellar tract, called the anterior spinocerebellar tract, which is illustrated here in red on the right side, but of course there is a red anterior spinocerebellar tract on the left side as well, but it's just not colored and illustrated. In this case, the information enters the spinal cord. There's the dorsal root ganglion. The information enters the spinal cord, whether we're at the cervical or the lumbar region, whether we're talking about arm proprioception or leg proprioception, enters the spinal cord in the dorsal horn. In the case of the red, the anterior spinocerebellar tract, the first synapse is in the dorsal horn. It crosses over, goes up the anterior spinocerebellar tract. And here's the funny thing. It crosses over again in the pons, in the brainstem. So it crosses once in the spinal cord, and it crosses again in the pons, and the information then synapses ends up in the ipsilateral, crossover, ipsilateral hemisphere of the cerebellum. The other spinocerebellar tract is the posterior spinocerebellar tract, illustrated here in blue on the left side, but of course there's one on the other side as well. Information from the legs or from the arms about position from the spindles enters the spinal cord in the dorsal horn, synapses in the dorsal horn, and the secondary axons go up they don't cross over, they go up the ipsilateral posterior spinocerebellar tract. They don't cross over, and the information ends up in the ipsilateral. Well, so here there's a double cross in the brainstem. But it also ends up in the ipsilateral. So the spinocerebellar tract doesn't cross. There's a double cross in the brainstem, and it ends up in the ipsilateral spinocerebellar tract. What do you need to know? You need to know that there is a spinocerebellar tract, that there is an anterior and a posterior part of the spinocerebellar tract, that the spinocerebellar tracts are very lateral. We'll cartoon that in another slide. They are even more lateral than the anterolateral column. And that the information from the spinocerebellar tract goes to the ipsilateral cerebellum, ipsilateral hemisphere of the cerebellum. You don't have to know where the crosses are, at least for me, for the spinocerebellar tract. You go to medical school, you deal with all of these very complex white and gray matter things in the brainstem. You do have to know, but not today, not this month. Questions about the spinocerebellar tract? more proprioception. So this is a slide for reference. Let's just illustrate what you know now. So this is a cartoon of the spinal cord. Lumbar, let's just say it's thoracic, somewhere in the middle. This is the right dorsal column, and that's the left dorsal column. And it carries ipsilateral touch, pressure, and position information. I'm just going to say this is the dorsal column, and there's the information that it carries. The anterolateral column this is the right anterolateral column, also called the spinothalamic tract, carries contralateral pain and temperature information because there was the crossover of the neuron that went up the anterolateral column. Just cartooning. The information. The arrows are trying to illustrate this is going to the brain. And the spinocerebellar tracts carry position information. There's the posterior spinocerebellar tract and the anterior spinocerebellar tract. So posterior spinocerebellar tract and the anterior spinocerebellar tract. CST. Carry ipsilateral and contralateral position information to the ipsilateral cerebellum. So we have some, I'm not going to cartoon it because it will just get too complicated. Remember the neurons have their cell bodies in the dorsal root ganglion. They enter the dorsal horn. Some of them, they all synapse in the dorsal horn. Some of them cross over and then go up. Some of them don't cross over and then go up. That's why some of it is ipsilateral and some of it is contralateral. Questions about proprioception? Or any of this? So as I said before, the information, whether it's carried by the pain and temperature information in the anterolateral column, or the touch pressure information carried in the dorsal column, or the position information carried either in the dorsal column or in the spinocerebellar tract, is going to synapse in the thalamus on the contralateral side, or in the ipsilateral cerebellum, depending, and end up, and then get processed further in the contralateral somatosensory cortex. So we're going to the somatosensory cortex now because the thalamus is too complicated. There's information processing in the thalamus. Don't forget the thalamus, an information relay network. But it's complicated. Let's go to the somatosensory cortex. So this is the part of the brain that's integrating information. Is it painful? No. Is it touch? Is it dangerous touch that's going to become painful? No, it's just touch. Is it a nice touch? Oh, let's go to the limbic center and activate all kinds of reward pathways. The brain is processing this information. Is it information about my body that is stretched? Is my body like this, or is my body like this? Okay, so all that information that's coming from your body is going to get processed in the brain. The somatosensory cortex is in the post-central gyrus, which is the most anterior gyrus of the parietal lobe, because the central sulcus is the separation between the frontal lobe and the parietal lobe. This is information you need to know. The somatosensory cortex is in the post-central gyrus. So this is looking at the brain from the top. So this is left and this is right in this image. So here they've taken a sort of a semi-oblique coronal section through the brain here at the level of the anterior level of the somatosensory cortex, the postcentral gyrus. So now we're seeing the postcentral gyrus in coronal section. Everybody clear on the orientation? We're taking the right somatosensory cortex and looking at it coronally. There's all kinds of good stuff here, like lateral ventricle, temporal lobe, etc. But we're going to focus on the postcentral gyrus of the parietal lobe. The body is mapped. The location of the information coming from the body to the brain is mapped locally on the somatosensory cortex. So information from the head and face, jaw, tongue, and throat is mapped laterally, the lateral part of the somatosensory cortex. And you can see that we get a lot of information from our face, and that takes up a lot of real estate, neuronal real estate on the somatosensory cortex. We get relatively little information from our guts, and that takes relatively, our whole digestive tract gives us some information, but it's mapped into a very small section of the somatosensory cortex. So our face and our hands get a huge chunk for processing, because those are the most important inputs to our somatosensory brain. So face, tongue, jaw, mouth, laterally. Hand, superior lateral. Arm, head, torso, superior. Leg and foot are on the medial. They're part of the cortex facing the longitudinal fissure. And the genitals are medial. So do I expect you to be able to draw the complete map? No. But I do expect you to know that face and mouth are lateral, hand and arm are superior lateral, body is superior, and legs are medial. I'll say that again in case you didn't catch it all. Face, tongue, throat, lateral. Hand and arm, superior lateral. body, superior, legs and genitals, median. Yes?
Did you say for the gut it's lateral?
The gut is lateral. I probably won't test you on that. So let's finish the somatosensory bit with a discussion of pain and pain modulation, or analgesia, pain inhibition. So here's a painful stimulus. Where are these nerve endings? In the skin somewhere. A painful stimulus to the skin. A acid-sensitive ion channel, or an ATP-sensitive ion channel, receptor, activating, depolarizing this neuron to threshold, action potentials. What is this supposed to illustrate? I want you to interpret this cartoon. What is this right? That's the dorsal root ganglion. That's the cell body in the dorsal root ganglion of this neuron. So being able to interpret, being able to transfer information from a cartoon like this, where it's clear, okay, this is now the classic cartoon of a dorsal root ganglion, to something like this, where some of the information has been stripped away, that's still the dorsal root ganglion. This is so where are we here? Where is the first synapse of the pain pathway? Dorsal horn. We're in the gray matter of the dorsal horn. Neurotransmitters are released. This slide is illustrating a peptide neurotransmitter called substance P. I haven't talked about it, but it's one of those protein neurotransmitters. Some of these neurons also release glutamate. Activates the secondary neuron. This is a secondary pain pathway. Does it cross over in the gray matter of the spinal cord? Pain does. Pain crosses over to the spinal cord. So this neuron, this cartoon, is leaving out crossover. What part of the spinal cord is this axon going up in the contralateral spinal cord? Anterolateral. So this is the axon is in the anterolateral pathway on the other side of the spinal cord from where the pain was coming in. Okay? So being able to transfer your information to a simple cartoon like this is important. And then there's a synapse in the thalamus and a synapse in the somatosensory cortex, and we say ouch. We feel pain. Saying ouch is a whole other thing. Now you know that you can take painkillers, and some of those painkillers, like aspirin or Tylenol or ibuprofen, are inhibiting this stimulus. They're inhibiting the inflammation that is causing the pain. So those painkillers are working out here. You also know that there are other types of painkillers, like opioids. Opioids are excellent painkillers. They have a very negative side effect of causing addiction, but they are excellent painkillers. And they work by activating presynaptic receptors, presynaptic inhibitory receptors on the first synapse. So we have neurons from the brain that release endogenous opioids, endorphins and enkephalins. Or you can take drugs that activate these receptors. So this is the endogenous pathway. Opioids and endorphins activate these presynaptic inhibitory receptors, or oxycodone, or what is Demerol, the drugs that you can be prescribed for pain, also activate these presynaptic receptors. These receptors, these opioid receptors, they have a name, they're called the mu opioid receptors, are GPCRs. What type of GPCR? So you see they are, this is inhibitory. What type of G protein do you suppose the mu opioid receptors are coupled to?
Yeah.
GI. Inhibits adenylyl cyclase, inhibits the calcium channels that would trigger neurotransmitter release. The beta-gamma subunit activates the G protein coupled inward rectifier potassium channels, hyperpolarizing the presynaptic terminal, inhibiting the release of the neurotransmitter, and inhibiting the information that gets to the pain. That gets the pain to the brain. Presynaptic inhibition by mu opioid receptors. Whether we're talking about an endogenous opioid, this is the wrong word, opioid, or an exogenous opioid like morphine or oxycodone or heroin. Now you probably, okay, so a receptor. So this morning I was realizing, wait a second, I'm going to be talking about a GPCR. I can't just leave it like that, little blob on a screen. No, that's not what I wanted to do. This is what I wanted to do. It's a receptor. Its structure has been determined back in 2018. Here's the receptor in green. This must be the membrane here. Here's the alpha, beta, and gamma. What do we see here? We see, so this is getting at one of the questions from the exam. It's a G protein coupled receptor. Transmembrane alpha helices is a structural motif of GPCRs. 7 transmembrane alpha helices is a structural motif. A binding site for opioid molecules is a key structural motif of the mu opioid receptors. An interface that interacts specifically with the GI alpha subunit. Doesn't interact with GS or GQ. It interacts with GI. It's a specific structural surface. So those are some of the structural information. And you can see, this paper was all about this illustration. This is going back to self-is. Here are some of the key residues that define the binding pocket. Here are different opioids sitting in the binding pocket. Here are different conformations of one of the opioids. Damgo is an experimental opioid. There's other figures that show conformational changes of the receptor, etc. But remember that when I show a cartoon of a receptor that has a blobogram on it, there's a lot more than just a blob. Right? That's the point I'm trying to make. Okay, let's do a couple of clickers. I got a whole bunch of them, and we won't have time to do all of them. Let's do a few, and then you can do the rest of this homework. In the somatosensory system, which of the following is a true statement? Different sensory modalities are detected by different types of receptors. All somatosensory information is detected by long receptor cells. Lateral inhibition is stronger in the touch vibration pathway, sensory afferents, than in the pain sensory afferents. Each part of the skin is innervated by the same number of sensory nerves. So I'll give you a hint. There's more than one correct answer. So which of these are true? Most of you are getting some of it right. So you all got A. That's true. They're all long receptor cells. All of these cells, the afferent has an axon. So in the somatosensory system, all of the primary afferents are long receptor cells. Right? Let me go back and illustrate. Receptor, axon, cell body, axon. This is a long receptor cell. Long receptor cell. They're all like this. Whether it's pain or touch or pressure or proprioception. Lateral inhibition is stronger in the touch and vibration sense. We can detect fine touch better. If you have a cut on your finger, your whole finger hurts, whereas the cut, because of poor or large receptive fields for pain. Receptive fields for pain are bigger than receptive fields for touch and pressure information. Each part of the skin is innervated by the same number of sensory nerves? No, we've demonstrated that the fingertip and the lip and the mouth are innervated by a large number of neurons in order to give us fine sensation, fine discrimination. And the back of the neck is innervated by fewer cells. They have larger fields, fewer cells. Okay, let's do the second one. In the spinal cord, which of the following is true? Axons of sensory neurons enter the spinal cord in the dorsal horn. All nerve cell axons are in white matter. Specific regions of the white matter carry information from specific sensory modalities. Sensory information from the body enters the spinal cord at different levels. Different levels of the spinal cord, cervical, thoracic, etc. Okay, we ready? Let's see. I think most of you are getting it right. A, C, and D are correct. Any questions about either of these clickers? So, again, the goal is not to test you. The goal is to answer questions. Yeah.
Could you re-explain C at the first one?
C, this is, I didn't teach this, but you probably know that your ability to detect a painful stimulus is less localized than your ability to detect a single touch, even on your lip. If you have a sore on your lip, your whole lip feels sore. In part it's because the inflammation spreads, but in part it's because we have larger receptive fields for pain than we have for touch and pressure. And so larger receptive fields means less lateral inhibition for pain. So I'm flipping it. Smaller receptive fields for touch and pressure mean that there's more neurons innervating it and they have sharper receptive fields, so there's better lateral inhibition. Did that help? Other questions? Yes?
So when a part of your body like has to be numbed for like the surgery or something,
Uh-huh.
is it just blocking the receptors?
Well, it depends on the drug that's being used.
Okay.
So if they give you, so let's do this. Let's go back here. So they can give an epidural, which is an injection of the drug into the spinal cord, and it's blocking this information here. It depends. Well, again, it depends on the drug that's being injected. If they inject an opioid, they're blocking the neurotransmitter release at the level of information. They can also, so let's just say they, instead of injecting it in the lower body, they can inject, say, here, and they can inject a drug that blocks the sodium channels. why would blocking sodium channels here be as effective as blocking neurotransmitter release here? Well, the information has to get past here. Information going is an action potential. If we block the sodium channels, we block the pain right here. So for example, my wife had to have arm surgery on she broke her elbow. So rather than, they sedated her with another drug that's in her, affecting awareness. She wasn't asleep, but she was sedated. And then they went to a ganglion in her shoulder and injected a very potent, long-lasting sodium channel blocker that basically paralyzed and numbed her entire arm for hours. So it wasn't local. Well, it was local, but it was local of the whole arm by blocking all information coming from the arm to the spinal cord. And that was a sodium channel blocker. And the anesthesiologist was very skillful at finding exactly where to inject this very potent sodium channel blocker in order to block, well, he was blocking this nerve. He was blocking action potentials in this nerve by hitting where the ganglion was. So you can block the pain in a number of ways. If you've ever had dental work where you need a Novocaine or some other painkiller, they are finding the ganglion, trigeminal nerve ganglion in your jaw, injecting a sodium channel blocker and preventing the pain information from getting to your spinal cord. That's a sodium channel blocker. They might give you a long-lasting opiate afterwards, because these are more orally available, in order to block the pain later. But they're being very careful about prescribing opioids now because of addiction. So we can block this pathway. We can block the pathway with anti-inflammatory drugs. We can block the pathway with sodium channel blockers. We can block the pathway with opiates or opioid-like molecules, or sodium channel blockers, or those are pretty much the way we block pain. Yeah?
So if you got a cortisone shot, it would block at the pain stimulus?
Cortisone is an anti-inflammatory, and so it is mostly acting by preventing the inflammatory signal from being initiated. Yes?
So can you there's people that have more analgesia.
Yeah,
others.
so
Can
some
you,
people are really
activate
good at
that?
this.
You can activate it by thinking about it, or?
Some people have learned how to, all of us can, in a crisis, we can activate this and not feel pain for some period of time. Some people are better at it, some people have trained themselves to do it more cognitively, more consciously, but we can do this, we do this all unconsciously as well. Yeah, so this pathway can be more robust in some people and less robust in other people. Yeah. Opioids are still the gold standard for pain inhibition, for analgesia in many settings. So they're the best. They last long, you can take them orally. You don't have to inject them like you do with a sodium channel blocker. There are other drugs that are used to inhibit this synapse, calcium channel blockers that block the calcium channels rather than activating the opioid receptors. But again, they have to be injected into the spinal cord. So, if you're in chronic pain situation, if you want to go to the hospital every day or to the doctor's office every day or every 2 days to get your shot, or do you want to just take a pill? Well, obviously taking a pill is better, except for the fact that it causes addiction. So it's a trade-off. Yeah.
Being able to without any type of medication, feel that pain, is that similar when you're in a high intensity situation, like a game, for instance, and you might get hit really hard or a cut and you don't even know?
Exactly. So this pathway can be activated by the sympathetic nervous system as well as by these sort of intrinsic mechanisms. So the sympathetic nervous system can activate this pathway. Fight or flight. when you're fighting, or even when you're fleeing, you're activating the pain inhibition pathway. Let's get the hell out of here. Other questions? Okay, so we did 2 of these clickers. I've got a bunch more. Let's not do them. Let me assign them to you as homework. The clickers and the answers are on Moodle, or they will be at the end of the class today. So I recommend that you go through all of these. Plenty of ways of self-testing, but do that on your own so that we have time to get started with the visual system. Oh, isn't that great? Okay, so let's take a short break here and get ready to do the visual system. What would happen if I had a drug that specifically activated, go back. Would a drug that specifically activated this neuron be a good painkiller? Yeah, it would be a great painkiller. But I can't imagine a drug that would be specific for this type of neuron. But that sort of thing. So people have been asking me, how can I prepare better for the next test? Do a lot of what-if questions. What if I were to inhibit action potentials here? What would happen? Well, get analgesia. What would happen if I cut my arm off? It happens. What would happen if my arm got cut off? And this neuron is chronically activated. The stump of this neuron is now hypersensitive and chronically activated. Well, that would be phantom limb syndrome. Because the limb is not there, but the neuron is saying, I hurt there. That's phantom limb. So that kind of thing. What would happen if I were to cut this pathway? Well, okay, maybe I feel no pain, but maybe, cutting a specific pathway is kind of tricky. What if it also damaged my touch and pressure and motor pathways? Well, now I can't feel anything and I can't move my body. So, what if questions? What would happen if I changed something? Just to be clear, drugs like morphine are excellent painkillers, but they have other effects in the brain, not at the spinal cord, but in the brain, which is what causes addiction. And so they are not excellent drugs because of the addiction side effects. Okay, I'm going to skip a bunch of clickers, but I do recommend going through them. Here is a overview. So we're changing modalities big time here. We're going into the visual system. So we're going to figure out how the retina transduces photons into electrical information that our brain interprets as an image. So I'm forming an image, but it's really just photons bouncing off of you and the rest of the room and hitting my retina, and then the nervous system happens. So we're going to talk about the retina. The information gets from the retina to the brain by the optic nerve, which is not shown here because it's been cut. We'll talk about what happens at the optic chiasm. We'll talk about the thalamus, the lateral geniculate nucleus of the thalamus. We'll talk about the neurons that take information from the lateral geniculate nucleus of the thalamus to the visual cortex in the posterior occipital lobe. So that's a big, fat overview of where we're going. Okay, so let's do some eye anatomy. Image. Just, physicists love to just use an arrow for their image, so that's what we did. There's an image. Photons coming from the image to our eyeball. The outermost window to the eye, really, is the cornea. It's a clear sheet of cells. It's cellular, transparent. Cornea. Behind the cornea is the anterior chamber, and it is filled with a fluid called aqueous humor. aqueous humor. Behind the anterior chamber, filled with aqueous humor, is the iris. The iris is a small muscle with a gap in it. It's a circular muscle, and the center of the circular muscle is a aperture called the iris. Called the pupil. So the iris is a circular muscle, so this is in cross section, and the center of the iris, where there is no muscle, is the pupil. The iris can, it's a circular muscle. It can dilate and it can constrict. When it constricts, less light gets through the pupil because it's smaller, and when it dilates, more light gets through the pupil because it's bigger. So this is the iris. My fingers are the iris. Constricting, dilate. Circular muscle. It's a sphincter. Constricting. You like that word? Yeah. Constricts or dilates. The pupil is the hole in the middle of the muscle. Behind the iris, behind the pupil, is the lens, another cellular structure that is transparent. The lens is supported behind the iris by tiny little ligaments called zonular fibers. So the zonular fibers are holding the lens in place, and they are connected to another sphincter muscle called the ciliary body or the ciliary muscle. The ciliary muscle is another circular muscle. So the iris and the ciliary body are circular muscles. The iris controls the diameter of the pupil, and the ciliary muscle controls the tension on the zonular fibers, which puts tension on the lens. So when the ciliary muscle is dilated, the tension on the lens is great, and the lens is flat. When the ciliary muscle constricts, the tension on the lens, on the zonular fibers decreases because the muscle constricts. The tension on the fibers decreases, and the lens rounds up. And this is going to be important for our ability to focus the light from either near or far objects. We change the shape of the lens to do focus. But the light is focused on the back of the eye where the retina is. So the retina is cartooned here as this red layer. We're going to break this down big time into the cellular structure of the retina. There is a section of the retina called the fovea where our receptive fields are best and where in humans we have color vision. So the fovea is where we tend to focus our gaze when we are focused on an object. So right now I am focusing on Tynan's nose, right? That is the center of my focus. So the image of Tynan's nose is focused on my fovea. All the rest of this image, when I'm focused on Tynan's nose, is outside of the fovea. If I'm getting visual information. I know that there's things over here, and I know there's things over here, and up here and down here. I can see them, but that's not where my attention, that's not where my attention is focused. I'm focusing on that image on my fovea. The peripheral vision is outside of the fovea. Okay? There is a blind spot, you've heard of it. The blind spot is right here. It is near, but it is not the same as the fovea. So the blind spot is not the fovea. The fovea is where we have our best vision, and the blind spot is where we have our worst vision. and I'll show you why we have a blind spot. It's where axons from the retina go out the back. So there are no photoreceptors here. So retina on the inside of the back of the eye, then a layer called the pigment epithelium, which is a nutritive layer that helps the retina continue to detect photons. And then behind the pigment epithelium, so retina, pigment epithelium, choroid, and then the sclera. The sclera is the white of the eye, the outer layer of the eye, connective tissue. Will I expect you to know this? Yeah, everything on this slide. Except I'm not going to ask you about visual axis, except for this. You can cross these words out. But everything else. Oh, I didn't talk about vitreous humor. So we have aqueous humor, and I will, even though it's not on here, you should label your slide with aqueous humor here and vitreous humor here. They're both clear solutions. Aqueous humor is watery, vitreous humor is like a gel. We're going to see that in lab next week. Okay, so there's a cross-section of the eye. Yeah.
So with the ciliary muscle, when it makes the lens more flat or more round, like which one is focused and which one is out of focus?
I'll show you in a couple slides. Question? You have time?
Can you talk about the rectus tendon?
I'll tell, next slide. Okay, so yes, you will have to know what the rectus is, but let's go to the next slide. There are muscles that control the movement of the eye. You know that. So let's talk about the muscles. The superior rectus is the muscle that attaches to the top of the eye. So that's what this tendon is supposed to illustrate, the superior rectus. And this would be the, you could label this would be the, what? Inferior rectus. So the superior rectus muscles attach to the top of the eye, to the superior surface of the eyeball, sclera. The inferior rectus muscles attach to the inferior surface of the eye, sclera. And so you can predict what these muscles are good for. They're good for elevating and depressing the eyeball, right? I can look up. I don't know, can you see me looking up? Or you can see me looking down. When I look up, I am contracting my superior rectus and relaxing my inferior rectus. And opposite for looking down. So the superior and the inferior rectus are important for elevation and depression, and looking up and looking down of the eye. We have the lateral rectus, which attaches to the lateral surface of the sclera, and the medial rectus, which attaches to the medial surface of the eye on both sides. So these are important for looking to the right or to the left. So when I look to the left, I am contracting the left lateral rectus of the left eye and the medial rectus of the right eye, right? I'm moving my left eye laterally and I'm moving my right eye medially. So we have this circuit for keeping our eyes focused on the same thing. We're not like chameleons where we can move our eyes around, any which way. We keep our eyes focused on the same thing. Some of you may have if you've ever looked at the eyeballs of a newborn baby, it's pretty creepy. Their eyes are wobbling all over the place because they have not yet learned how to keep their eyes really well moving in parallel. So moving the eyes to the left, so this is from the top, so this is the left. Moving the eyes to the left would be a contraction of the left lateral rectus and a contraction of the right medial rectus. Moving the eyes to the right would be a contraction of the right lateral rectus and the left medial rectus. We also have oblique muscles, which are pretty tricky. We have a superior oblique and a inferior oblique that are important for subtle rotational movements of the eyes. We can rotate our eyes to keep the images upright. They're tricky, but the point is we have them, and so you do have to know the name of them. Superior and inferior rectus, medial and lateral rectus, superior and inferior oblique. So this is the bone of the nose. This structure right here, that through which the superior rectus goes, I'm sorry, the superior oblique muscle goes through this structure called the trochlea. Trochlea is Latin for pulley. And you can see why. Okay, so I will want you to know the names of these muscles and what they do. The oculomotor nerve innervates most of these muscles. So what are the exceptions? So this is a cartoon of the oculomotor nerve. So what does the oculomotor, I know this is complicated, let me try to simplify this. This is focusing on the oculomotor nerve. The oculomotor nerve innervates the superior oblique, the superior rectus, and the inferior rectus. It also innervates the medial rectus. The oculomotor nerve innervates the superior rectus, the inferior rectus, and the medial rectus. It also innervates through a ganglion the iris and the ciliary muscle. So the constriction of the iris and our ability to focus is a function of the oculomotor nerve. That's not a conscious thing. So some of this is intentional, right? I elevate my eyes, I'm using my oculomotor nerve in a conscious way. I go into a dark room, I dilate my pupil in an unconscious way. But that's both oculomotor nerve. What else did I want to say about this? The nerve that innervates the superior oblique is the trochlear nerve. The superior oblique goes through the trochlea, the pulley, and it is innervated by the trochlear nerve. The lateral rectus, on both sides, is innervated by the abducens nerve. Abducens, abduction. The lateral rectus is important for abducting the eyes. That's how you can think of it. Abducens, abduction of the eyes is a function of contracting the lateral rectus. Yeah?
Why can't we contract both lateral rectus?
Our body, we have, so if you've ever looked at a newborn, they can. Their eyes, they can go like, but we have developed synapses that just say, if you're gonna move your right eye to the right, if you're gonna abduct your right eye, you're gonna adduct your left eye. We're just gonna do it like that. We're not gonna develop the neural pathways of that. We can cross our eyes, I can go cross-eyed, but I can't easily abduct my eyes on both sides. Yeah.
Can you quickly reiterate which nerve again?
Okay. The oculomotor nerve innervates the superior rectus, the inferior rectus, the medial rectus, oh, and, I forgot to say, and the inferior oblique. And the iris and the ciliary muscle. The trochlear nerve innervates the superior oblique. Trochlear nerve, superior oblique. There's the trochlea right there in this cartoon. The abducens nerve, which is not illustrated here at all, innervates the lateral rectus. Okay? So now I'm going to get to, I think it was Thalia's question, or question over here. Thalia, were you asking about focus? Let's talk about focus. Distant object. So I'm focusing my eyes on, what am I focusing on? What's the name of that door? I'm blanking on my memory. Bundy. Focusing on Bundy. It's a distant object. Distant object. I'm putting the image of Bundy on my retina. I'm using my eyeglasses as assist devices to help me focus because I couldn't do it without my glasses. But with my glasses, I can focus that image on my retina. And focusing on a distant object needs a flat lens. The flat lens means the ciliary bodies are putting a lot of tension on the zonular fibers in order to flatten this lens. So for me to focus on a distant object, my oculomotor nerve is telling my ciliary body to relax. It's not activating my ciliary body. It is letting it relax, put tension on the fibers, flatten the lens. But now I'm changing my focus from Bundy to my fingertip. And I'm focusing, this is my focus now. I'm focusing very close. Focusing on a close object, putting that image in focus on my retina requires a round, bulgy lens. In order for my lens to bulge, I need to constrict the ciliary muscle. So the oculomotor nerve is constricting the ciliary muscle, putting less tension on the zonular fibers, allowing the lens to round up, and putting the object, the image of the object in focus on my retina. So when I focus on a distance, I am relaxing my ciliary muscles. And when I focus on a near object, I am contracting my ciliary muscles. And I'm doing it unconsciously. Unless I say, okay, I'm going to put my gaze here, but I'm really going to focus back here. Okay? Yeah?
So how does like contracting release tension?
Contracting releases tension on the object that is in the middle of it. Does that help?
Yeah.
Contracting puts less tension on these fibers. Constricting puts more tension on the fibers. I'm sorry, relaxing. So constricting puts less tension, allows for a round lens. Constriction puts more tension, makes the lens flat. Yeah?
I'm confused with how like both in focus and out of focus without relaxed ciliary muscles.
Well, so if I have a relaxed ciliary muscle and I'm focusing on a distant object, the object is in focus on my retina. If I have so right now I am focusing on Bundy. My fingertips are right there, it's right in my fovea, but it's not in focus. If the lens is not bulgy, if the lens is flat, the image of the near object is not in focus on my retina, and so it's blurry. In order to make this object in focus on my retina, I have to bulge my lens. In order to bulge my lens, I have to constrict the ciliary body and allow the lens to bulge. Okay, let's stop here. There's a whole bunch of clickers related to the stuff that I've taught you, so a whole bunch of clickers with answers. And what we'll do after, which is homework, because I won't go through them on Thursday, we'll start with how the retina turns photons into signals, electrical signals that will get into this. This is where we'll start.